promising new saRNA vaccine for COVID-19 fight, but questions remain. Exciting news is emerging in the battle against COVID-19 with the development of a novel type of mRNA vaccine, self-amplifying mRNA, saRNA. While questions remain, this technology holds significant promise. The results are published on Business Wire article on February 18, 2024. This article discusses the results of a study conducted by Meiji Seika Pharma in Japan. The study shows that saRNA vaccines offer longer-lasting immunity compared to conventional mRNA vaccine boosters. Let's first understand how traditional mRNA vaccines work. They deliver a snippet of the virus's genetic code to our cells, instructing them to create harmless copies of a viral protein, like the spike protein. These copies trigger our immune system, building defenses that recognize and fight the real virus if encountered. saRNA vaccines take this concept further. They include a special enzyme that multiplies the viral protein copies within cells, potentially boosting the immune response. This approach offers potential benefits: lower dose, compared to traditional mRNA vaccines, Moderna/Pfizer, 30 to 100 micrograms, saRNA requires only 5 micrograms, potentially reducing side effects and production costs. Easier storage, freeze-dried format eliminates stringent refrigeration needs, making it ideal for regions with limited resources. Longer protection. Early data suggests saRNA vaccines might offer more extended protection against COVID-19. However, important questions remain. Efficacy. While the saRNA candidate showed 95% effectiveness in preventing severe illness and death, its 55% effectiveness against symptomatic COVID-19 falls short of initial mRNA vaccines, 90 to 95%. Variants. Testing against newer variants like Omicron BA.4/5 is ongoing, with the current data reflecting effectiveness against the original Wuhan strain and Delta variant. Long-term effects. Like any new vaccine, long-term studies are crucial to ensure safety and sustained effectiveness. But before we further digest the clinical results of saRNA vaccine, in the following 2 diagrams, I would like to review how a self-amplifying RNA vaccine works, and then moving on to conventional mRNA vaccines. Let's begin with a molecule known as the self-amplifying RNA. This RNA, labeled from 5-CSE to 3-CSE, is a complex structure containing the CAP, NSP1-4, target protein, poly-A, and 3'-CSE. The next phase of our journey takes us to the amplification of this RNA. The amplified RNA enhances the translation process. It's depicted that the RNA is converted, negative RNA strand is turned into a positive strand facilitated by RDRP enzyme. We then move on to in situ translation. Here, a cellular machine known as the ribosome translates the self-amplifying RNA into target proteins and the RDRP enzyme. This is a critical step, as it means that the RNA is translated into protein within the cell. Finally, we arrive at our destination, the target proteins. These proteins can be used as antigens for a vaccine or as a therapeutic modality. It's important to note that these are at an early stage of research and development, R&D. Now, let's transition to conventional mRNA vaccines. The process begins with a conventional mRNA that contains the CAP, 5'-UTR, target protein, poly-A, and 3'-UTR. This mRNA is represented as a purple rectangular shape in our diagram. The next phase is in situ translation, where a ribosome translates the conventional mRNA into target proteins. This means that the mRNA is translated into protein within the cell itself. The ribosome, which is the site of protein synthesis, is represented as a green circle in the diagram. Finally, target proteins are produced, which can be used as antigens for a vaccine. These proteins are represented as a multicolored shape in the diagram. It's specifically mentioned that these proteins act as antigens for vaccines, such as the COVID-19 vaccine. Our diagrams use various shapes and colors to represent different elements like the mRNA, ribosome, and proteins involved in the process. Arrows indicate the flow and steps of the process. These visual representations help us to understand the complex processes of how self-amplifying RNA vaccines and conventional mRNA vaccines work. There are one Science and one Nature paper published about saRNA vaccine, as this slide summarizes. Nature.com, published on December 6, 2023, this article explores the potential uses and benefits of saRNA vaccines that have been researched for over 20 years. It highlights the first full approval of an saRNA vaccine. Science.org, published on April 8, 2022, this article discusses the first self-amplifying RNA, saRNA, granted full regulatory approval anywhere in the world. The new vaccine platform could provide defenses ranging from various infectious diseases and conceivably possibly other major health concerns. And because it could be used at a lower dose, it might have fewer side effects than other messenger RNA, mRNA, treatments have. These articles provide a comprehensive exploration of the first approval of saRNA vaccines and their replication and expression strategies. For more detailed and accurate information, I recommend referring to these original sources. Despite these questions, saRNA represents a significant step forward. It holds the potential to make vaccines more accessible, easier storage and distribution benefit resource-limited regions. Affordable, lower production costs could translate to wider affordability. Efficient, lower doses and potentially longer protection could streamline vaccination regimens. This technology extends beyond COVID-19, offering promise for vaccines against other infectious diseases. Researchers are actively studying saRNA, and continued advancements are anticipated. While it may not immediately replace existing vaccines, the possibilities offered by saRNA are encouraging. Remember, staying informed and following public health guidelines remain crucial in our fight against the pandemic. As we wrap up this episode, thanks for tuning in. Stay curious, and we'll catch you in the next episode of Biology of Pharmaceuticals channel. Please like, subscribe, and share if you would like to support this work. We will see you next time. Have a nice day.